Hello everyone, here we are going to discuss about some germ cell tumors and their macroscopic and microscopic views. First we will discuss about seminoma. Here you can see a macroscopic view of seminoma. When we talk about the appearance of a seminoma, you can see it is a soft and well defined tumor and it is grey white in color. You can see we call this surface as cut potato appearance. Here cut potato appearance is there, and you can see it is homogeneous and well circumscribed. Although this is a malignant tumor, it has a well circumscribed and homogeneous appearance, so it is an exception from normal malignant features. Here is a microscopic view of a seminoma. You can see in microscopic view there are lobules which is divided by fibrous septae. Here fibrous septae are there. Here there are lymphocytic infiltrations in the fibrous septae, and the cells are polyhedral or polygonal in shape. Here you can see, and also they have well demarcated margins here, and prominent nuclei and nucleoli also there. Here also again another microscopic view of seminoma. You can see fibrous septae is there and lymphocytic infiltrations in fibrous septae. It has made up of collagens and it is eosinophilic. You can see lobulated structures here, and also there are cells with clear pale cytoplasm and well demarcated borders, and they are polyhedral in shape, and there is a nice rounded nuclei in the center like features. Next we will discuss about teratomas. Teratomas are neoplastic germ cells that differentiate along multiple somatic cell lineages. They can be have uh, elements that are derived from all three germ layers. That means the structures that derived from endoderm, ectoderm, mesoderm you can see in these teratomas. There are two types of teratomas as mature cystic teratomas, which is the benign tumor, and immature teratomas, which is the malignant formation. Mature cystic teratomas, which is the benign tumor, and immature teratomas, which is the malignant type tumor. Here, this is a mature cystic teratoma. Here, it is a cystic mass which is filled with sebaceous material. You can see here it is like sebaceous material. Here some hair like structures are there. Here this is a tooth like structure. I told you previously in a teratoma there can be any structure which derive from endoderm, mesoderm, ectoderm can be seen in a teratoma. So there are hair like structures, tooth like structures, that means bones, cartilages, any of the material can be there. We call this as matted hair. So this is a mature cystic teratoma, which is the benign tumor. Here also the same picture, mature cystic teratoma. Here is a microscopic view of mature cystic teratoma. You can see various tissues, adipose tissues, sebaceous glands, and hair follicles, as well as keratinized stratified squamous epithelium. Here is another picture with high power. Here you can see cartilages and fat. Here whitish color, fat, and here cartilage. Here is a picture of immature malignant teratoma. Immature malignant teratoma, macroscopic view. Here you can see it is a bulky tumor, not like mature cystic teratoma. It is cystic mass, while this is a bulky tumor. This has solid cut surface and areas of necrosis. Here you can see there are areas of necrosis and hemorrhages, and it has solid cut surface. There are immature elements or minimally differentiated cartilages, bones, muscles, nerves, or other tissues as well. Then we will talk about gestational trophoblastic diseases. That means they are abnormal proliferation of fetal trophoblastic cells. Gestational trophoblastic diseases can be divided into two groups as molar lesions and non-molar lesions. What are the two groups? Molar lesions and non-molar lesions. We call molar lesions as hydatiform moles. These molar lesions can be partial, complete, or invasive. That means partial hydatiform mole, complete hydatiform mole or invasive hydatiform mole. And there are non-molar lesions like choriocarcinomas. All of these tumors can release HCG, and they can be identified by HCG in blood or urine, and they are higher than normal pregnant values as well. Here, first we will talk about hydatiform mole. That means molar lesions. Here, this is look like a bunch of grape appearance. You can see nicely. Actually, these are fluid-filled vesicles. These are fluid-filled vesicles which look like bunch of grapes. These fluid-filled vesicles means the expanded chorionic villi. These are the expanded chorionic villi, look like fluid-filled vesicles or bunch of grape appearance. When there is a hydatiform mole, the uterine cavity is distended like in pregnancy, but it is too large for date in pregnancy. It is too large for the date. Here is also another picture of hydatiform mole. Here is the microscopic appearance of complete mole, and there are hydrophic degeneration and distended villi. Here in this picture you can see distended villi with hydrophic degeneration. In complete hydatiform mole, there are no fetal parts, but in partial hydatiform mole, there are fetal parts. You have to remember that. Here is a partial hydatiform mole microscopic appearance. There is only a proportion of villi shows edema. Only some villi shows edema. But in complete hydatiform mole, all the villi are distended with hydrophic degeneration. Here is a choriocarcinoma, which is a non-molar lesion, comes under gestational trophoblastic diseases. In this choriocarcinoma, you can see extensive necrosis and hemorrhages can be seen. Chorionic villi are not formed in choriocarcinomas, and it has a very high potential to become metastasized. Here you can see a microscopic view of choriocarcinoma. Here from this picture you can get an idea about complete hydatiform moles, partial hydatiform mole, choriocarcinoma, invasive moles. Here complete hydatiform mole, there are no fetal parts. Uterine cavity is completely filled with those villi. Here in partial hydatiform mole there are fetal parts can be seen. Here invasive mole has invaded the uterine wall. Here you can see it has invaded the uterine wall, and here this is a choriocarcinoma. This is a choriocarcinoma. So that's all about germ cell tumors. Thank you for watching.